So it's been over 20 years since Jamie Thompson derived the first embryonic stem cell line, and over 10 years since he and other researchers generated the first human-induced pluripotent stem cells from human fibroblast cells. And so much hype was generated from these achievements, mainly because these cells can divide indefinitely, but also because they can form the 200 plus specialized cell types of our bodies. And so because of these achievements, much hype much promise was gained from these cells to develop stem cell therapies for many diseases and injuries that currently have no cures or no adequate treatments. And so many mice models have been developed, and that's a big problem. And so mainly because we're not mice. And so curing a disease in a mouse model doesn't necessarily mean that you can treat or cure a disease in humans. And so we need better models to be able to translate these discoveries into human therapies. Many researchers have developed and utilized, expensive non-primate human models, but these models, besides being expensive, they do not, because we don't share the same environment together, we obviously don't naturally have the same diseases and injuries as these models as well. There is a better model, and that's the dog. Okay? The domestic dog has more naturally, besides humans, have more naturally inherited diseases than any other species. Because of similar physiology, disease progression, and clinical response, the dog, at least over half of all canine disorders, have human equivalents. And so we're using the dog model as a way to translate human stem cell therapy into the clinic. And so we are among some of the first groups in the world to derive canine embryonic stem cells many years ago. These cells exhibit the same characteristics as these human counterparts. They have prolonged cell division, and they can form any cell type of the body. And so my lab, including colleagues here at Western, at the Ontario Veterinary College, and also across Canada, are devising ways to take these cells into the clinic. We're devising bioreactors to amplify these cells into therapeutic doses. We are optimizing protocols to make clinical grade patient-specific stem cells for transplantation therapies in the vet clinic, but also using these cells and genome modifying them so that we can make cell models of disease. And so when we can make these cells from concept to cure in this clinically relevant animal model, we can thus enable these cells to be able to treat not only our best friends, but our humans as well.